Yes, go ahead, Vincent. You raise your hand. Yeah, so immunotherapy. Oh, okay. So okay. All right. So you drop now, right? So we won't be having the report on COVID-19 and cytokine storm next week. So I would like, okay, let me again share my screen, no, so that I'll will. Okay, thank you for that, no. So it means that next week we will be. Can you see the screen? Okay, it's still loading there. So next week we will be hearing the report this time of Gem and Sol because okay, let me put it here now, no. Uh, crap. Okay, so let's all right highlight. Okay, so next week, please prepare Gem and Sol. So you'll be reporting on principles in kidney transplantation. All right. Okay, noted. Okay, Yulyung, the floor is yours. Vincent, are you still raising your hand? No more. Okay, all right. Go ahead, Yulyung.
Can you see that?
Yes, yes, we can see. We can see. Thank you.
Okay.
Hello everyone, I'm Laura. I will present the topic is about immunotherapy. Okay, next page. Let's look at the content. The first one is immunomodulators. Second one is activation immunotherapies. The third one is immune enhancement therapy. The next one is suppression immunotherapies. The last one is hemetic therapies. Okay, next page. Let's look at what is the definition of immunotherapy. Immunotherapy or biological therapy is the treatment of disease or by activating or suppressing the immune system. Immunotherapies designed to elicit or amplify an immune response are classified as activation immunotherapies, while immunotherapy that reduce or suppress are classified as suppressing immunotherapies. Okay, next page. The diagram above represents the process of chimeric antigen receptor T cell therapy.
I'm sorry to butt in, Yung Sora. Can we move to the next slide? Yeah. Okay.
Sorry.
That's okay.
Have some problems.
Yeah, we will wait. No problem.
I start over. Can you see that?
Not yet. Not yet.
Okay.
Maybe still loading. Yes, we can see now. Thank you, Sora.
Okay.
Hello everyone, I'm Zora. I will present the topic is about immunotherapy. Okay, next page. Let's look at the content. The first one is immunomodulators. Second one is activation immunotherapies. The third one is immune enhancement therapy. The next one is suppression immunotherapies. The last one is hematologic therapies. Okay, next page. Let's look at what is the definition of immunotherapy. Immunotherapy or biological therapy is the treatment of disease or by activating or suppressing the immune system. Immunotherapies designed to elicit or amplify an immune response are classified as activation immunotherapies, while immunotherapy that reduce or suppress are classified as suppressing immunotherapies. Okay.
Sora, sorry to interrupt again. Your slides are not moving. Maybe you should click the slideshow and then press enter. Vincent, do you have a copy of the slide of Sora? Can you control that? Or okay. So this is slide number 3 now. Okay, go ahead. Go ahead.
Hello? Professor, Zora recorded the slides before the class because
Yes, okay.
She's not very good in English, so she recorded it first. Okay, let me present the slides first.
All right. Okay, you can share your screen, Vincent.
Hello everyone, I'm Zora. I will present the topic is about immunotherapy. Okay, next page, let's look at the content. The first one is immunomodulators. Second one is activation immunotherapies. The third one is immune enhancement therapy. The next one is prevention immunotherapies. Another one is hemispheric therapies. Okay, next page, let's look at what is the definition of immunotherapy. Immunotherapy or biological therapy is the treatment of disease or by activating or suppressing the immune system. Immunotherapies designed to elicit or amplify an immune response are classified as activation immunotherapies, while immunotherapy that reduce or suppress are classified as suppressing immunotherapies. Okay, next page. The diagram above represents the process of chimeric antigen receptor T-cell therapy. This is a method of immunotherapy, which is a growing practice in the treatment of cancer. The final result should be a production of equipment T-cells that can recognize and fight the infected cancer cells in the body. The first one is T-cells are removed from the patient's blood. Then in a lab setting, the gene that encodes from the specific antigen receptors are incorporated into the T-cells, and thus producing the receptors on the surface of the cells. The newly modified T-cells are then further harvested and grown in the lab. After a certain time period, the engineered T-cells are infused back into the patient. Okay, next page. Immunomodulators. Immunomodulators are the active agents of immunotherapy. They are a diverse array of recombinant, synthetic, and natural preparations. Okay, next page. It's about activation immunotherapies. Cancer, main article, cancer immunotherapy. Cancer treatment used to be focused on killing or removing cancer cells and tumors with chemotherapy or surgery or radiation. These treatments can be very effective and in many cases are still used. In 2018, the Nobel Prize in Psychology or Medicine was awarded to James Allison and Tosco Hanjo for their discovery of cancer therapy by inhibition of negative immune regulation. Cancer immunotherapy attempts to stimulate the immune system to destroy tumors. A variety of strategies are in use or are undergoing research and testing. Randomized controlled studies in different cancers, resulting in significant increase in survival and disease-free period have been reported, and its efficiency is enhanced by 20 to 30% when cell-based immunotherapy is combined with conventional treatment methods. One of the oldest forms of cancer immunotherapy is the use of Bcg Vaccine, which was originally to vaccinate against tuberculosis and later was found to be useful in the treatment of bladder cancer. Bcg immunotherapy induces both local and systematic immune responses. Some mechanisms by which Bcg immunotherapy mediates tumor immunity have been widely studied, but they are still not completely understood. The use of monoclonal antibodies in cancer therapy was first introduced in 1997 with rituximab, an anti-CD20 antibody for treatment of B-cell. Since then, several monoclonal antibodies have been approved for treatment had methodological malignancies as well as for solid tumors. The extraction of GCSF lymphocytes from the blood and expanding in vitro against a tumor antigen before re-injecting the cells with appropriate stimulatory cytokines. The cells then destroy the tumor cells that express the antigen. Topical immunotherapy utilizes an immune enhancement cream, which produces interferon causing the recipient killer T cells to destroy warts, actinic keratosis, basal cell cancer, vaginal intraepithelial neoplasia, and cell cancer. Cutaneous lymphoma and superficial malignant melanoma. Injection immunotherapy uses MAPS, Candida, and the HPV cancer tracing antigen injections to treat wounds, HPV-induced tumors. Adoptive cell transfer has been tested on lung and other cancers, with greatest success achieved in melanoma. Okay, next page. It's about dendritic cell-based pump priming or vaccination. Dendritic cells DC can be stimulated to activate cytotoxic response towards an antigen. Dendritic cells are a type of antigen-presenting cell harvested from the person needing the immunotherapy. The cells are then either blasted with an antigen or tumor site or transfected with a viral vector, causing them to display the antigen. Again. So first, FDA approved cell-based immunotherapy, the cancer vaccine. Spillocer-T is one example of this approach. The Immune Response Cooperation IRC developed this immunotherapy and licensed the technology to Dendron, which obtained FDA clearance. The current approaches for this based vaccination are mainly based on antigen loading in vitro generated DCS from monocytes or CD34 plus cells, activating them with a different TLR ligands and cytokine combinations and injecting them back to the patients. Then in level targeting approaches comprise administrating specific cytokines and targeting the DCS with antibodies to say type lectin receptors or agonistic antibodies that are conjugated with antigen as interest. Future approach may target disease subsets and based on their specifically expressed type lectin receptors or chemokine receptors. After a potential approach is a generation of genetically engineered DCS from induced pluripotent stem cells and use of new antigen loaded DCS for inducing better clinical outcome. Okay, next page. T-cell adaptive transfer. Adaptive cell transfer in vitro cultivates autologous extracted T-cells for later transfusing. Alternatively, genetically engineered T-cells are created by harvesting T-cells and then infecting the T-cells with a retrovirus that contains a copy of the T-cell reporter TCR gene that is specialized to recognize tumor antigens. The virus integrates the receptor into the T-cell's genome. The cells are expanded non-specifically or stimulated. The cells are then reinfused and produce an immune response against the tumor cells. And the technique has been tested on refractory metastatic melanomas and advanced skin cancer, the first FDA-approved CAR-T drug. Using this approach to obtain the clinical and commercial supply of the CAR-T, Novartis purchased the manufacturing plant. The distribution system and hired the production team that produced developed by Dan Jordan and the Immune Response Corporation. Whether T-cells are genetically engineered or not, before infusing, lymphodepletion of the recipient is required to eliminate regulatory T-cells as well as unmodified endogenous lymphocytes that compete with the transferred cells for homeostatic cytokines. Lymphodepletion may be achieved by monoablative chemotherapy, to which total body radiation may be added for greater effect. Transferred cells are multiplied in vivo and persisted in peripheral blood in many people, sometimes representing levels as 75% or CD8 plus T-cells at 6 to 12 months after infusing. As of 2012, clinical trials for metastatic melanoma were ongoing at multiple sites. Clinical responses to adoptive transfer of T-cells were observed in patients with metastatic melanoma resistant to multiple immunotherapies. Okay, next page. It's about checkpoint inhibitors. Main article, checkpoint inhibitor, anti-PD-1 or PD-L1 and anti-CTLA-4. And boys are the two types of checkpoint inhibitors currently available to patients. The approval of anti-cytotoxicity lymphocyte-associated protein for CTLA-4 and anti-program so that protein 1 PD-1 antibodies for human use has already resulted in significant improvement in disease outcomes for virus cancers. Although these molecules were originally discovered as molecules playing a role in T-cell activation or apoptosis, subsequent preclinical research showed their important role in the maintenance of peripheral immune tolerance. Immune checkpoint inhibitors are proved to treat some patients with a variety of cancer types, including melanoma, breast cancer, bladder cancer, cervical cancer, and colon cancer, head and neck cancer, or Hodgkin lymphoma. These therapies have revolutionized cancer immunotherapy as they showed for the first time in many years of research in metastatic melanoma, which is considered as one of the most immunogenic human cancers, and important improvement in overall survival with an increasing group of patients benefiting long-term from these treatments. Okay, next page is about immune enhancement therapy. Autologous immune enhancement therapy use a person's own peripheral blood-derived natural killer cells, cytotoxic T lymphocytes, cellular cells, and other relevant immune cells are expanded in vitro and then reinfused. The therapy has been tested. The therapy has been tested against hepatitis, chronic Fagin syndrome, and HHV6 infection. Okay, next page it is about suppression immunotherapies. Immune suppression dampens an abnormal immune response in autoimmune diseases or reduce normal immune response to prevent rejection of transplanted organs or cells. Immunosuppressive drugs help manage organ transplantation and autoimmune disease. Immune responses depend on lymphocyte proliferation. Okay, cytostatic drugs are immunosuppressive. Glucocorticoids are somewhat more specific inhibitors of lymphocyte activation, whereas inhibitors of immune finds more specifically target T lymphocyte activation. Immunosuppressive antibodies target steps in the immune response. Other drugs modulate immune responses and can be used to induce immune regulation. It has been observed in a preclinical trial that regulation of the immune system by small immune suppressive molecules such as vitamin D, and the examethasone and curcumin administered under a low dose regimen and subcutaneously could be helpful in preventing or treating chronic inflammation. Okay, the next page is about immune tolerance. The body naturally does not launch an immune system attack on its own tissues. Models generally identify CD4 plus T cells as the center of the autoimmune response. Loss of T cell tolerance then unleashes B cells and other immune effector cells onto the target tissue. The ideal tolerogenic therapy would target the specific T cell coordinating the autoimmune attack. Immune tolerance therapies seek to reset the immune system so that the body stops mistakenly attacking its own organs or cells in autoimmune disease or accept foreign tissue in organ transplantation. Recent therapeutic approach is the infusion as regulatory immune cells into transplant recipients. The transfer as regulatory immune cells has the potential to inhibit the activity of the effector. Creating immune tolerance reduces or eliminates the need for lifelong immune suppression and attendant side effects. It has been tested on transplantations, rheumatoid arthritis, Taiwan diabetes, and other autoimmune disorders. Okay, the next page, you can see the two charts. And the next page is about the ologies. And the main article is ology in immunotherapy. Immunotherapy can also be used to treat allergies. While allergy treatments treat allergic symptoms, immunotherapy can reduce sensitivity to allergens, lessening its severity. Immunotherapy may produce long-term benefits. Immunotherapy is partly effective in some people and ineffective in others, but it offers allergy sufferers a chance to reduce or support their symptoms. The therapy is indicated for people who are extremely allergic or who cannot avoid specific allergens. IgE-mediated food allergy is a global health problem that affects millions of persons and affects every aspect of life for the patient. Promising approach to treat food allergies is used as oral immunotherapy, OIT. OIT consists in a gradual exposure to increasing amounts of allergen can lead to the majority of subjects tolerating doses of food sufficient to prevent reaction of accidental exposure. And those ages increase over time as the person becomes desensitized. This technique has been tested on infants to prevent peanut allergies. Allergy-specific immunotherapy, ASIT, has become the gold standard for the quantitative treatment for IgE-mediated allergic diseases for a large variety of allergens. One may curiously await the new developments which will further enhance our understanding of allergy mechanisms and improve ASIT for the next generation of patients and physicians. Okay, the next page is about hemiphagic therapies. Wormborne and hookworm have been tested for immunological diseases and allergies. Hemphagic therapy has been investigated as a treatment for relapsing remitting multiple sclerosis, Crohn's, allergies, and asthma. The mechanism of how the hemicells modulate the immune response is unknown. The hypothesized mechanisms include repolarization of the Th1 and Th2 response, and the modulation is dendritic cell function. The hemicells regulate the pro-inflammatory cytokines, interleukin 12, IT12, interferon gamma, IF, and the tumor necrosis factor alpha. While promoting the production regulator, the second, and the cytokines such as IL, such as IL-10, IL-4, IL-5, and IL-13. The co-evolution with hemicells has shaped some of the genes associated with interleukin expression and immunological disorders such as Crohn's, ulcerative colitis, and celiac disease. Hemicells' relationship to humans as hosts should be classified as a mutualistic or symbolic. Okay, that's all as my representation.
Thank you so much, Sora, for that. Okay, so the floor is open for some questions to Sora. I think we can entertain one or two. Okay, this is pre-recorded. All right, no problem. Okay, so do you have some questions to Sora? Anyone? Okay, Sora, just a quick question lang, no? So basically, you mentioned that immunotherapy is intended for patients like having cancer, di ba? Yeah, I was wondering, would you know what would be the difference between a immunotherapy over chemotherapy? Are they the same? Or may difference? Can you compare and contrast between a immunotherapy over a chemotherapy? What's the main difference between the two? All right. So basically, when we say immunotherapy, no, usually these are biologicals. And when we say biologicals, they are actually produced in the laboratory through recombinant DNA technology. No, we use living organisms to manufacture these proteins, including, of course, your MABs. These are your monoclonal antibodies. On the other hand, when we say chemotherapy, these are synthetically produced, no, chemically synthesized. No, they are not biologicals. So that's the main difference between the two. The other one, biological in nature, no, and the other one is chemically synthesized. Okay, All right. So, do you have some questions to Sora before we proceed to the next reporter? I think Alex is already here, no? So he will be reporting about developments of vaccines. Okay, so yeah, let's hear naman from Alex. Alex?
Okay, Doc. Turken will share his screen po.
All right, Alex.
Okay. Is my voice clear?
Yes, sir.
Yes, very loud.
Mic test. So anyway, thank you. Good evening, Dr. Buckley. Good evening, everyone. Sir Alex and I were going to discuss about development of vaccines. So our report will be divided with the following. First is what are vaccines? Second, principle of vaccination. Third, vaccine ingredients and development. Additionally, packaging, storage, and shipping will also be tackled. We will also talk about the current vaccine technologies and novel vaccine technologies, and lastly, the challenges in vaccine developments. So, vaccinology is regarded as one of the greatest successes in the field of medicine. Through vaccination, several once feared, debilitating, and deadly diseases in history are currently either controlled, eliminated, or eradicated. All these thanks to collaborative work of researchers around the world, advancement of science and technology, and aspiration of improving the lives of all people. So, what are vaccines? These are biological suspension of weakened or killed pathogens or their components. It is a form of immunoprophylaxis, which means prevention of disease through immunization. Apart from being an immunoprophylactant, vaccines are studied as potential cure rather as a form of immunotherapy for cancer patients, which we will note later what type of vaccine it is. So the principle of vaccination lies that the vaccine simulates active immunity and create an immune memory so that exposure to the micro, active disease microorganisms will stimulate an already primed immune system to fight the disease. In this illustration, we can see the effect of vaccines in the immune system. Traditional approach creates immune memory, and the next time the body is attacked by some pathogen, a specific learned response will be generated by adaptive immunity. Memory T cell will destroy infected cells, whereas the memory B cell generates antibodies, bind targets to block infection as well as targeting pathogen. On the other hand, when trained immunity is harnessed, this enhances non-specific responses by innate immune system, such as the monocyte and neutrophils once diverse pathogens infects. The responsiveness and protection against the pathogens are enhanced. Now let's go to vaccine ingredients. Vaccines are made up of several components, namely antigen, adjuvant, preservatives, stabilizers, surfactants, residuals, and diluents. Antigens are the primary and active component of vaccines since it generates an immune response. Antigens create the blueprint for making the active component. Antigens may be a small part of the disease-causing organism, like a protein or sugar, or it may be the whole organism in a weakened or inactive form. Preservatives, on the other hand, prevent the vaccine from being contaminated once the vials has been opened, if it will be used for vaccinating more than one person. 2-phenoxyethanol is the most commonly used preservative which can be found in baby products. Another preservative used in multi-dose vials is thimerosal. Since thiomersal, the other name for thimerosal, is a mercury-based preservative, this preservative is considered a regulated substance. Thereby, there is a currently reduced usage of thimerosal as a vaccine preservative. There are some countries also who banned the usage of thimerosal as mercury as a preservative for vaccines. Surfactants keep all the ingredients in the vaccine blended together, and by doing so, this prevents settling and clumping of elements that are in the liquid form of the vaccine. The action of surfactant is that it lowers the surface tension of the liquid. Example of surfactants is polysorbate 80. The residuals are tiny amounts of various substances used during manufacturing or production of vaccines that are not active ingredients in the completed vaccines. So these residuals depends on the process used, may come from cell culture mediums, egg proteins, or other animal-derived ingredients, culture cells, yeast, antibiotics, or by inactivating agents such as formaldehyde. Next is a diluent are liquid used to dilute a vaccine to the correct concentration immediately prior to use. The most commonly used diluent is sterile water. Lastly, we now have adjuvant, which is a significant component of vaccines. Adjuvants improve the immune system to the vaccine sometimes by keeping the vaccine at the injection site for a little longer, or by stimulating local immune cells. In other words, adjuvant increases antibody titers and induces cell-mediated immunity. There are three distinguished types of adjuvants. These are aluminum salts such as aluminum phosphate, aluminum hydroxide, and potassium aluminum sulfate, oil in water emulsions, and microparticles. Aluminum salts has been used in a number of vaccine formulations, particularly in the vaccines against hepatitis A virus, hepatitis B virus, human papillomavirus, diphtheria, tetanus, meningococcus, pneumococcal conjugates, and Haemophilus influenza type B. Now let's head to the development of vaccines. These are the stages of vaccine development and testing. As the candidate vaccine makes its way through these processes, the regulation and oversight also increases. Typically, developing vaccines usually takes 10 to 15 years, and most of these years are spent on the clinical phase. So the exploratory phase of vaccination. In exploratory phase of vaccine development, this involves basic laboratory research and often lasts 2 to 4 years. Federally in this stage, researchers and scientists identify natural or synthetic antigens that might help prevent or treat a disease. These antigens could include virus-like particles, weakened virus or bacteria, weakened bacterial toxins, and other substances derived from the pathogens. So during the clinical phase, this vaccine formulation is first tested in animals such as mice and monkeys to assess its safety and potential to prevent disease. Apart from animals, tissue culture or cell culture systems might also be utilized during this phase. The purpose of this phase, apart from safety, is to assess the immunogenicity of the vaccine for researchers to have an idea of the cellular responses they might expect in humans. During this phase, the safe starting dose for the next phase of research and safe method of vaccine administration will be determined. Since animals are utilized in this study, researchers in this phase may opt to do challenge studies. This means that the vaccinated animal will be intentionally infected with the pathogen, which again, in order to assess the vaccine candidate. So preclinical stages often last 1 to 2 years. So once the safety and immunogenicity of candidate vaccine has been determined, the candidate vaccine will be subjected to the clinical phase. However, prior to that, sponsors particularly located in the United States, sponsors are the private companies who manufacture vaccines, are required to submit an application for investigation new drug to the US FDA. So in that application, the manufacturing and testing processes will be described, laboratory reports are summarized, and the proposed study is also described. Clinical protocols must be approved by an institutional review board. This stage usually takes 30 days. So after it is approved, it is given a go signal to do the clinical phase. So in the phase 1 of the clinical phase, the candidate vaccine is given to a small number of volunteers to assess its safety. confirm it generates an immune response, and determine the right dosage. This phase involves a small group of individuals. So phase 1 trials may be non-blinded or open label. This means that researchers know what treatments are being given to each study subject or human or experimental group. The human subjects in this study know as well the treatment that they have received. So researchers may use the challenge model by which participants are attempted to be infected with a weakened or modified version of the pathogen. Participants in the study are carefully monitored, and as well as the conditions are carefully controlled. So once the candidate vaccine passed the clinical phase 1, phase 2 clinical trials follows up, by which the candidate vaccine is given to several hundred volunteers. Multiple trials are conducted, and some individuals in the clinical trial may belong to groups at risk of acquiring the disease. The trials are randomized and well-controlled and includes a placebo group. So a placebo group are individuals who are given an inactive substance that seems identical to the vaccine being tested. In the phase 3 of clinical trials, the candidate vaccine is given to thousands of volunteers and are conducted across multiple countries and multiple sites within a country. The trial is randomized and double-blinded, which means that neither the human subject and researchers know who got the vaccine. The experimental vaccine is pitted against a placebo, which may be a saline solution or a vaccine for another disease. The purpose of phase 3 clinical trials is to assess vaccine safety in a large group of people and to detect certain rare side effects that have not surfaced in the smaller groups of subjects. Tested in earlier phases, vaccine efficacy is tested as well. So once the candidate vaccine passes all 3 clinical trials, vaccine developers will submit a biologic licensure application to the US FDA. So the FDA will inspect the factory where the vaccine will be made and approve the labeling of the vaccine. Aside from inspection, the FDA will continue to monitor the production of the vaccine as well as conduct its own testing of manufacturers' vaccines. After approval and manufacturing and distribution, the approved vaccine will be monitored. This includes phase 4 trials, vaccine adverse event reporting system or VAERS, and vaccine safety data link or VSD. So vaccine companies may conduct phase 4 trials after a vaccine is released. This is an optional study where the vaccine is continued to be tested for safety, efficacy, and other potential uses. Another monitoring system is the vaccine adverse event reporting system, in which this is a voluntary reporting system whose goal is to detect possible signals of adverse events. These adverse events will be investigated by the CDC and tries to find out whether the adverse event was caused by the vaccine. Apart from monitoring adverse events, data from VAERS would be used to identify potential patient risk factors, identify vaccine lots with increased numbers or types of reported adverse events, and lastly, assess the safety of newly licensed vaccines. The Vaccine Safety Data Link, on the other hand, is a collection of linked databases that contain information from large medical groups. Researchers can access the data by proposing studies to the CDC and having them approved. During manufacturing, vaccines are made in bulk quantities and may either be lyophilized or liquid form. These are bottled in glass vials, since glass vials are durable and able to maintain the integrity of the vaccine even in extreme temperatures. So vaccines are required to be stored in cold temperatures. Thus, this involves the use of specialized equipment. Ordinary refrigerants should not be used, such as the regular refrigerators at home should not be used since the temperature cannot be monitored in these conditions. Most vaccines are stored in refrigerated temperature at 2 to 8 degrees Celsius, such as Bcg Vaccine and Tdap. AstraZeneca vaccine for COVID-19 is also stored in this temperature. There are vaccines that require as cold as negative 20 degrees Celsius, such as Mrna Vaccine. Mrna Vaccine requires to be stored at negative 15 to negative 25 degrees Celsius. Then there are vaccines that needs to be stored at ultra cold temperature at negative 70 degrees Celsius. Example of this is the Pfizer vaccine for COVID-19. So during shipping, the vaccines are shipped using specialized equipment from manufacturing plant. Once a vaccine arrives at our country or country of destination, refrigerated lorries are utilized in order to transport the vaccine from the airport to cold storage warehouses. From there, portable ice boxes are employed to take the vaccines from the cold room to regional facilities. In case vaccination takes place outside these regional facilities, portable ice boxes would be used to transport the product to local areas. So let's head now to novel vaccine technologies, which will be discussed by Sir Alex.
Hello. I'm actually going to discuss about current vaccine technologies, and this will encompass live attenuated vaccines, inactivated vaccines, and subunit vaccines, and some of mRNA vaccines, which are relatively new but have been studied for decades. Next slide, please. So vaccines are basically antigen preparations that are administered to prevent infectious diseases. Next slide, please. So let's begin with live attenuated vaccines, which have been routine since Edward Jenner's smallpox vaccine, which, if you remember your undergrad studies, is based on cross-reactivity, wherein cowpox virus was used to develop immunity against an antigenically similar but highly pathogenic smallpox. However, the problem is that most pathogens do not have immunologically similar but less pathogenic counterparts. So if we head to the next slide, our solution will be to modify the bacteria and or viruses so that they will lose the pathogenic properties but are still capable of stimulating a good immune response. And one way to do that is to culture them under conditions different from those present in the host and are unfavorable for its growth. So next slide, please. Thank you. So an example of this is when Louis Pasteur used non-pathogenic strains of chicken cholera and attenuated rabies virus from dried rabbit spinal cords that were infected with rabies. Another example is the Bcg vaccine, which was actually done by Albert Calmet and Camille Guerin. It's a tuberculosis vaccine from attenuated Mycobacterium bovis grown in culture media with increasing bile concentrations. So what happened was Mycobacterium bovis adapted to growing in high bile concentrations and was therefore suitable for the human body because the human body contains low bile concentrations. And so it is not conducive to pathogenic growth, but it will still induce an immune response. Next slide, please. Another example is typhoid fever, which is a vaccine that has mutated Salmonella typhi. It was chemically treated to induce genetic mutations that weaken them and make them no longer pathogenic. Next slide, please. Thank you. Live attenuated vaccines are more easily prepared against viral infections than bacterial infections. And an example of this is our next slide, which is polio. Polio vaccine, polio, if you remember, it causes septic meningitis, which is very detrimental to those infected. So in 1960, Albert Sabin made an oral polio vaccine from a live attenuated strain, which he cultured in monkey kidney cells containing 3 types that are capable of causing disease. However, this vaccine was replaced. This was actually the vaccine used from 1963 to 1987 until an inactivated polio vaccine was licensed. This is because 1 per 2.7 million cases of those with the oral polio vaccine had paralytic polymyelitis. So in third world countries such as the Philippines, this is still the routine because it's more easily accessible. However, there is still some risk, even though very little, still some risk. I actually experienced this. If I don't know if your barangays have the patak patak, it's that one. So next slide, please. I also have our MMR vaccines, which are short for measles, mumps, rubella, and sometimes it's MMRV, which also includes varicella. This is actually prepared using repeated passage in culture cells. So measles are prepared in chick embryo cells, rubella in human diploid cells, varicella. They use a live attenuated oca strain from a Japanese child with chicken pox, sequentially propagated in cultures of human guinea pig cells. So next slide, please. There's also a very a more potent formulation of an attenuated oca strain, which is used for herpes zoster or shingles or the adult chicken pox. Okay. Next slide, please. We also have a live attenuated vaccine for influenza, which is administered intranasally. It has the hemagglutinin and neuraminidase antigens. However, flu vaccines are developed yearly because of the virus's high mutation rate. And how it is prepared is it is cultivated in chicken eggs and attenuated for adaptation for cold temperatures. So one advantage of live attenuated vaccines is that it is able to replicate at a low temperature in the host, and it's capable of inducing both humoral and cell-mediated responses. But the disadvantage is that it cannot be administered to immunocompromised people or pregnant women because mutations may occur. Remember our example in polio where the attenuated form of the polio virus returned to its pathogenic form. And of course, another disadvantage is the handling and storage, which may not be available in third world countries. This is because, remember, we are still dealing with a live pathogen. It's just weakened. So those are the limitations. Now we head to our inactivated vaccines, which are intact, killed viruses or bacteria. They are either killed by heat or chemical treatment, but still retain their antigenic properties. What is what is used usually for the killing of the pathogen is formaldehyde or beta propiolactone. This is used more than heat because heat alters the chemical structure of surface epitopes. Next slide, please. So an example is our classic influenza vaccine. We also have an inactivated influenza vaccine apart from uh, the attenuated one, which contains H3, H1N1, and one or two influenza B strains. The H3N2 and H1N1 are actually influenza A strains. They are grown in embryonated hen eggs and are killed by, of course, formaldehyde or beta propiolactone. And next slide, please. We also have a new influenza vaccine, which is grown in cell culture systems or recombinant hemagglutinin antigens. So this minimizes the use of eggs because some patients have allergic reactions to vaccines because of the use of certain proteins such as egg proteins. If I'm not sure if you're familiar with the news, but one football player in the NFL in the US refused to get the COVID vaccines because he was allergic to one of the ingredients that was posted in the WHO website. And therefore, he was fined for over $1 million. But the advantage of this is it is faster to produce. So next slide, please. We also have hepatitis A vaccine. To the right is an example of a hepatitis A patient that is having jaundice. So it comes from a purified hepatitis A, which is cultured in human fibroblasts and inactivated by formalin treatment. So next slide, please. And the advantage of an inactivated vaccine is that it can be safely given to immunocompromised people and it will not replicate in the host. However, for the disadvantage, a larger amount of antigen is needed to stimulate effective immune responses. Therefore, booster shots are needed. And of course, the vaccine, because it does not it it is already killed. The pathogen is already killed. Uh, it predominantly induces humoral immune response with little to no cell-mediated immunity. That's why we keep getting vaccine doses for such inactivated vaccine. We now head to subunit vaccines, which consists of one or more purified components of a pathogen that are capable of stimulating immune responses. So there are four that I will discuss: toxoid, polysaccharide, purified protein, and recombinant protein. So next slide, please. So a toxoid or toxoids are bacterial exotoxins chemically inactivated to not harm the host but retain their ability to stimulate an immune response. So these toxoids actually depend on the toxin that the bacteria will produce. So for Corynebacterium diphtheriae, there's formalin inactivated toxin. Clostridium tetani, there's also formalin inactivated toxin. And for Bordetella pertussis, which causes whooping cough, it has a cellular pertussis vaccine. So it's called capital DTAP, capital T, DAP, DT, TD. It's actually depends on the amount of diphtheria, tetanus, and a cellular pertussis component present. So next slide. So the polysaccharide vaccines. Polysaccharides, some bacteria have hydrophilic polysaccharide capsules that allows the bacteria to resist phagocytosis and other immune defenses. It will mask components of the membrane that might be immune response targets. And this is what we use in polysaccharide vaccines. So these vaccines contain multiple polysaccharide types because capsular antigens vary with different bacterial serotypes. And this will ensure that the immune response will provide a broad protection from the antigen or the pathogen, right? One example of this is Streptococcus pneumoniae, which contains polysaccharides from 13 to 23 serotypes. And Haemophilus influenzae also, which contains polyribosyl ribitol phosphate of HIV capsule conjugated to a protein carrier. Another is Neisseria meningitidis, which has 4 purified bacterial capsular polysaccharides. These are the A, C, Y, and W135. Next slide, please. However, a big disadvantage of polysaccharide vaccines is that they do not induce good immune responses because polysaccharides are T-independent antigens that stimulate IgM production with no immunoglobulin class switching or long-term memory response. So if you recall in your dengue, IgM is the first antibody to be produced and then it will switch its class to IgG, and then you will have a long-term memory response. In this case, the polysaccharide vaccines, that does not happen. So next slide, please. To remedy this problem, we use glycoconjugates, which are which is when polysaccharide antigens are linked to a carrier protein, such as tetanus or diphtheria toxin. This will induce a more effective immune response because this will activate T helper cells, and therefore it will result in immunological class switching, production of IgG, and memory cell generation. So it's very complicated. And another is purified protein vaccines, which are from the name just composed of proteins from a pathogen, purified proteins from a pathogen. And an example of this is the vaccine for Bordetella pertussis, which again causes whooping cough. So it's rare but serious neurological effect. It causes encephalitis or encephalopathy and convulsions in children with neurological disorders. So it contains 2 to 5 purified B pertussis toxins, one of which is a toxoid from pertussis toxin. So next slide, please. Now we had move on to recombinant protein vaccines, which are highly purified protein vaccines, which uses recombinant DNA technology. So what they do is that the gene coding for a specific pathogenic protein is isolated and incorporated into the genome of a non-pathogenic bacteria or yeast or other cell. This genetically modified cell is cultured in large quantities so that it will produce the antigen, and then the antigen is isolated. So an example of this is the Hepatitis B vaccine. So the HBs antigen is isolated from pooled plasma of infected patients. The HBs antigen is cloned in yeast cells, so to propagate it, and then it is harvested and purified. This purified protein will spontaneously assemble into virus-like particles, which are non-infectious but induce a very effective immune response. Next slide, please. Another example is the human papillomavirus, which contains a recombinant L1 major capsid protein from 2 to 9 HPV virus types. Which is highly associated with anal genital cancers. So these L1 genes are cloned in yeast cells as well, or insect cells infected with baculovirus, which is a virus that infects invertebrate cells. Next slide, please. So a very relatively new, but it has been researched for decades, is the mRNA vaccine. I was tempted to put a picture of the pioneer of mRNA vaccines, but I'm not sure if you're aware with the news, but he is very controversial for what he is saying about vaccination, especially in our sensitive time. So I'd rather not mention his name or endorse his face. Just know that if you search the pioneer for mRNA vaccines, that he's very controversial. So mRNA vaccines consist of mRNA strands inside a coating, and this coating protects the mRNA. The mRNA tells cells to make protein found in the antigen, and this protein will induce an immune response. Again, it does not contain a live virus and carries no risk of causing disease in a vaccinated person. So next slide, please. So one example that I chose was the COVID vaccine from Mrna and Pfizer. This is actually from Dr. Silverio's lecture in our introduction class to biochemistry, because I have a transcript of it. So use of a virus as a vector to insert mRNA of COVID spike protein. So remember, COVID is not recognized by our immune system because it it's it's theorized that it is a genetic spillover from an animal source such as the bat or the pangolin. But adenovirus is recognized by our body. Therefore, if we use adenovirus as a vector to insert the mRNA of a COVID spike protein, we can create a natural immune response and therefore uh, have an immunity to COVID. So, Sir Ken will continue with novel vaccine technology.
So, due to advancement in research and technology, new vaccine technologies are emerging. New antigens, viral vector-based vaccines, and virus-like particle-based vaccines are among the vaccine technologies developed. Neoantigens are non-autologous proteins with high individual specificity that is generated by non-synonymous mutations in the tumor cell genome. What makes neoantigens special is that it possesses stronger immunogenicity and higher affinity toward MHC and are not affected by central immunological tolerance. Thus, neoantigens are possible use in tumor vaccine therapy, and identification of a neoantigen is critical in that said kind of treatment. This is because neoantigens can not only enhance anti-tumor immune response, but have also reduced the risk of autoimmunity. Neoantigens can be recognized by CD8 plus and CD4 plus T cells in tumor tissues and thus trigger an anti-tumor response in vivo. In a study by Carreno et al., it was reported that dendritic cells loaded with neoantigens can trigger specific T cell responses in patients with melanoma. In order to identify neoantigens on tumor cells, whole exon sequencing, computer-simulated epitope prediction, and immunohistochemistry can be utilized as such. So in this illustration, the mutations in the tumor tissue produces neoantigens, and these clonal neoantigens can be expressed by a large number of proliferating cells. Through the use of software packages, the sequence of tumor cells and normal cells are compared. The sequences are contrasted and mutation neoantigens in tumor cells are identified. This leads to predicting the immunogenicity of antigen, and candidate neoantigens are evaluated and prioritized, after which a neoantigen vaccine will be designed. However, the use of neoantigen vaccines has limitations. Neoantigen vaccines cannot achieve complete elimination of malignant tumors. Thus, other therapies such as radiotherapy and chemotherapy are needed. So, another limitation is that the use of neoantigen vaccines is also limited by the diversity of somatic mutations in different tumor types and their individual specificity. Also, large differences among tumor types and individuals limit the use of tumor vaccines targeting mutated neoantigens. So, another novel vaccine technology is the viral vector-based vaccine, which relies on the delivery of one or more antigens encoded in the context of an unrelated and modified virus. This either employs live, replicating but often attenuated, or non-replicating vectors. So vaccine vector is engineered to encode for heterologous antigens that are shuttled into the host cells by the vector. Upon delivery, antigens are expressed and the host is able to induce immune responses against the respective target pathogen. So viral vector-based vaccine employs a wide array of different viruses such as adenoviruses, paromyxoviruses, toga viruses, rhabdoviruses, and pox viruses. So adenovirus vectors is the most commonly employed viral vector, and adenovirus-based vaccines can be constructed as replication-competent or replication-defective vectors, which are generated by replacing the E1A and E1B genomic region by an antigen expression cassette, thereby abolishing the viral ability to replicate. So the viral E3 and E4 genes are frequently deleted to prevent the elimination of adenovirus infected cells by the immune system and leaky expression of the inserted antigen. Adenoviral vectors are able to induce potent antibody as well as T-cell responses with variations in the immune response depending on the serotype employed. So, for example, replication deficient AD5 is able to induce exceptionally potent CD8 T cells as well as antibody responses. So the advantages of viral vector-based vaccines is that this represents a valuable and highly versatile platform for vaccine development. The viral genomes can be manipulated to express any antigen of choice, and the ability to stably accept relatively large insertions in the genome. Also, viral vectors induce stimuli in the target cells that mimic natural infection, thereby inducing potent immune responses. Viral vector-based vaccines can be delivered without additional adjuvants and with variations depending on which vector is employed. Strong antigen-specific cellular and humoral immune responses against the target antigen can be induced. So viral-based vector vaccines can be delivered or can be formulated without using more adjuvants. So, however, the disadvantages of viral vector-based vaccines is that they are genetically modified organisms. The use of viral vectors raises safety concerns for use in humans, also concern for potential reversion of the attenuated viral vector to virulence, especially when using a replication-competent vector, is one of the disadvantages. So each viral system requires different cellular systems for high-yield propagation, necessitating different manufacturing facilities for each viral vector platform. Viral vector-based vaccines are highly complex and comparatively cost-intensive process. So these are the examples of viral vector-based vaccines. So the examples are Oxford AstraZeneca vaccine, Sputnik 5, Janssen from Johnson & Johnson, and Convidencia vaccine. These vaccines are against COVID-19. They usually, uses viral vector-based vaccine technology. So another novel vaccine technology are virus-like particle-based vaccine. They use platforms capable of producing particles that mimic the structure of authentic viruses. They can be produced by expressing antigenic proteins in a eukaryotic or prokaryotic system, resulting in the formation of particles with an inherent ability to self-assemble. So alternatively, VLP vaccines can be made by producing blank VLP templates and then chemically linking antigenic peptides on the preformed particles. The VLP vaccines can fully activate immune systems of the vaccinated individual and are considered highly immunogenic and can stimulate robust cellular and humoral immune responses due to their highly repetitive display of antigenic epitopes. So these are the list of VLP vaccine candidates such as the Hepatitis B Vaccine. These are the name of the vaccine, Ingerix. The HPV vaccine, which market as Cervarix. HBV vaccine, Recombivax. And human papillomavirus vaccine marketed as Gardasil. So Sarah Alex will discuss the challenges in vaccine technology development.
Thank you, Sir Ken. Hello again. So let's start with the next slide. We have immunological challenges in developing a vaccine. Most of our vaccines are actually developed by determining components that consistently stimulate antibody responses in infected patients, often without detailed knowledge of immune mechanisms required for protection. A large number of our vaccines were simply formulated with aluminum hydroxide adjuvant and then injected. This is very problematic because if you go to the next slide, there is a broad agreement that the development of new and effective vaccines cannot proceed without a detailed understanding of the immune correlates of protection. However, this data is dependent on material from patients who recovered from natural infection and animal models. wherein animal models are very problematic because they must respond to infection and vaccine formulation in a very similar manner to humans, which is very problematic. Yeah. So if we head to our next slide, an example of this is the intranasally administered influenza vaccine, which contains a heat labile, enterotoxin, and E. coli adjuvant. It actually called Bell's palsy in humans, despite having strong safety in animals. This is because, in an example of an animal model is the mouse. Its immune system differs radically from humans. And the adaptive immunity, for example, its antibody classes differ. In humans, we have IgA1 and IgA2, which is very high in serum. However, in mice, there's only one class of IgA, which is very low in their serum. So you see, it's very hard. So next slide, please. So there has to be a well-controlled laboratory condition for animal studies. However, this still causes disparity when advancing work to human trials. So phase 2 trials often disappoint due to the target population having natural tendency for immune responses. This is because there is a high prevalence of chronic or immunosuppressive infections. Some volunteers in the phase 2 trials are very young or very old or have poor nutrition or obese, have pre-existing immunity to the vaccine vectors, and some of them have natural exposure to antigenically similar organisms to the pathogen. So, next slide, please. We also have a very big problem in antigen selecting methods, but the prevalence of bioinformatics and high throughput screening used for identifying new vaccine antigens for new and emerging pathogens for organisms dangerous or difficult to culture still does not have enough data to remedy this. For an example, again, if you head to the next slide. Sorry, I did not include it. Well, an example is the meningococcal antigen GN1870 because our presentation is very long-winded, so I did not want to bombard you with so much information. So anyway, this meningococcal antigen, it binds to human complement factor H and allows for the survival in the body. However, it is unable to bind to human complement factor H in rats or mice. So this led to human factor H transgenic mouse strains to further the vaccine research. But this is still, again, very difficult. This is one way that bioinformatics is used, but it's still very difficult. So selective pressures and antigenic variability of pathogens, improving antigenicity of non-protein antigens, weak immunogens, which cause a lag in availability of adjuvants and delivery systems to induce required protective immune responses. Next slide, please. There's also the problem of antibody response to surface molecules. Vaccines may be strain-specific and require multivalent formulation and have to be changed frequently. So, next slide, please. Ayan, weak immunogens are also a problem. What if the immunodominant antigen may not be protective when inserted into humans for immunological response, but instead it is only expressed as an immunological smokescreen? So therefore, we need to have more powerful adjuvants to activate immune cells such as B and T cells, macrophages, and dendritic cells to induce more protective levels of immunity. Next slide. This is why we have a new generation of adjuvants which activate innate immunity via pathogen-associated molecular patterns. So, very toxic na word. So these examples of this PAMP are bacterial cell walls, DNA with pattern recognition receptors or PRR, TNRs, C-type lectin, and many whole cell inactivated vaccines contain PAMP. So an example of this new generation of adjuvants is beta-glucan, which is rich in PAMP and stimulates C-type lectin, lectin-1, which is high in dendritic cells and neutrophils and macrophages. And another example is ISCO matrix. ISCO matrix, hindi po yung nagtutu joints at sumasayaw, which induces an innate immune cell-stimulating adjuvant, which induces a potent, long-lived, and protective humoral and cellular immune response. So next slide. This is one of the bigger concerns when talking about challenges in vaccine development. So vaccine research is usually for academia and not-for-profit organizations. However, vaccines are brought to market and developed by pharmaceutical companies. Therefore, the market cannot be ignored. However, the demand of vaccines does not coincide with the ability to pay. For example, third world countries which have high rates of diseases such as polio, they will not contribute to the monetary gain that pharmaceutical companies are eyeing. So, yeah. So next slide, please. There's also this problem that new vaccines are produced by a single manufacturer. Therefore, price competition is not a driver to reduce cost. And cost to benefit analysis may factor in for developed countries. So an example for this is in the US, there is chickenpox vaccination for all children. It is included in the childhood panel. However, in the UK, the United Kingdom, the NHS considers the impact on community. So the circulating virus from the chickenpox will boost immunity in adults and reduce the number of cases of shingles in older people. So the cost of treating increased number of adults with shingles is greater than treating mild illnesses in children. So, but the chickenpox vaccine in the UK is available privately. So next slide, please. There's also this problem. True vaccine costs for development are not disclosed by pharmaceutical companies, which is very problematic because sometimes companies claim to have high developing costs and cannot be interrogated if it's justified. This lack of transparency will impact the purchaser's informed decision making on reasonable, fair, and affordable prices. An example of this is a somewhat controversial topic again, wherein the Romanian member of the European Parliament revealed documents which were agreements that the big pharma company forced the governments around the world to sign. And if it was exposed on national TV that these documents were blacked out with black ink, so you can't read the information available. So it's a big problem. So next slide. We'd like to end with a quote that once a vaccine is in use, it must be continuously monitored to make sure it continues to be safe. Thank you. And our references are that. Thank you so much.
Thank you so much, Alexander, and of course, Kenneth. That was a very comprehensive discussion, but equally quite provoking and controversial nga. So I enjoyed that, no, actually. Thank you so much for that. Okay, so the floor is open for questions if you have one. All right, from start to end, no? It was very interesting and yeah, may mga punchline din si Kenneth and Alexander. I also enjoyed that too. All right, do you have some questions to Kenneth? Go ahead, go ahead.
Sir, Doc, I actually have
yes,
this
yes.
one is a very tagal na tanong na to since
Okay,
I was high school. Is there a possibility of developing a multi-potent vaccine? Like one vaccine for like all, not necessarily all, but for most
parang campaign na.
viruses.
Tetravalent.
Yes.
Alexander or Kenneth, do you like to answer that?
It's I guess in the after centuries, it's very hypothetical question, but as you can see, there are so many challenges in developing vaccines, starting with the immunological problem of different antigens and the way our body reacts and how different it is from testing it on animals and testing it on us. And apart from that is that how will we develop that? Because in remember there are different types of vaccines and some consist of live attenuated strains, some inactivated, some mRNA, and even more technologies are being developed. And if we put it into one vaccine, how will these interplay with one another? So I'd say maybe after centuries or when very new and very uh, uh, more developed technologies are available. We are limited by the technology of our time. Thank you.
Okay, thank you for that, Alex. Peter, would you like to ask questions?
I would say the inactivated one, sir, because the pathogen is killed. And remember, the live attenuated is weakened. And for the subunit vaccines, sometimes they consist of toxoids from the pathogen itself. So there are still some side effects. So I would say the inactivated. However, uh, you need multiple doses, as I have discussed, to keep being immune to the diseases that the pathogen brings. Thank you. I hope I answered your question. Uh, Fastest. I would I would say live attenuated because I can't say much on mRNA because it actually just rolled out. But I would say the fastest is live attenuated because remember it's the most potent and it causes both humoral and cell mediated immunity. So I would say live attenuated. You po- um I'm not entirely sure because remember research is still ongoing until now. And if you look at the different technologies that use I don't know, it's very difficult to say because vaccines are such a very difficult thing to produce. Like you can just imagine the cost, and apart from that, the controversy from producing, and also the clinical trials. And live attenuated vaccines are very dangerous because remember they're live vaccines just weakened, and so the clinical trials palang it would be uh, very taxing to research. But I think it's possible if we're staying on the hypothetical, and I think it's some of it is research. It just doesn't go past the clinical phase trials. Thank you. Mr. Ken ka naman magtanong.
All right, thank you so much for that, Peter, and of course, Alex, Kenneth, and Abdul Mahid. Okay, I know you're very, I don't know, hungry now for your dinner, but I just would like to share 3 or 4 slides now. No, I just like to add this as an additional input to what we have discussed, and also this is in line with what Abdul Mahid mentioned earlier. No, so actually, this is one of the promising tools that we have now because of the discovering of, let's say, for example, the Human Genome Project, and then, of course, the different tools ngayon available, no, like in genomics, in transcriptomics, no, looking also the proteomes and even metabolomes, and more recently, even looking at the vaccinomics. So there are a lot of omics, so to speak, no, from genomics to transcriptomics, proteomics, metabolomics, what have you. Anyway, what I'd like to highlight with this slide is that, as you can see here, no, these are the different approaches nga, the omics, and we have a lot of methods or techniques that are available naman, no, to continue to help identify candidates, no, for drug discovery, including vaccines then, no. So these are some of the important tools that we have now. For example, the availability of bioinformatics tools that will be able to data mine, no, do data mining, no, so that we can do in silico first, no, we can predict the, let's say, the 3D structure of a protein, whether it will fold or not, and so on and so forth. So once successful, then we can do na yung actual clinical trial. Aside from bioinformatics, of course, we can use, as mentioned earlier, yung animal models, which is the traditional one if you want to, let's say, do a functional study or functional analysis. And then when we talk about proteomics naman, no, we have the usual na mga Maldiv, no, 2D gel electrophoresis, and to name a few, no. And then you have also a page, no, 2-dimensional polyacromide gel electrophoresis as well. And then finally, when we talk naman about vaccinology, you have very powerful tools like your nuclear magnetic resonance, no, NMRs. So these are some of the promising tools available, no, to help identify future vaccine candidates, no. Maximizing the knowledge, the understanding about this genome to RNA to protein, and of course metabolome and vaccines. Now earlier na mention ni Alex yung egg base, no, and that was quite provoking, no, yung sabi niya yung isang athlete na NFL ba yon, na hindi siya na vaccine because of he is allergic to eggs, no. But as we all know, most of the vaccines are egg based, especially flu vaccines in particular, no. So this has long been used, and they are both used nga in inactivated, as mentioned by Peter, and also live attenuated vaccine. So I have here a photo which I borrowed. I just forgot to write the reference. So this is how they screen, no, yung mga eggs in terms of quality. So they put a candle onto the eggs and see whether this eggs would be a candidate to be used for later on vaccine development. The next slide, medyo toxic, busy, and I think we will need enough time to discuss this, but I will upload na lang sa TLC. But nonetheless, this is how it is being done, no? From the surveillance first, what are the emerging variants, so they will take into account that, and then of course looking at the prevailing strains also, no, from different locations using mga tools, no, in epidemiology. And then later on, yeah, they will start to inject this into the eggs, no, and come up with the vaccine. So more or less, this will be the flow, no, of how we develop vaccines, in particular influenza vaccines. And this is how it looks like, no, when done in an industry. So yeah, it will take about 10 to 12 days to inoculate this, no. And they will be putting in the eggs onto this machine and to check whether it's healthy or not, no, to screen for quality eggs. And then as mentioned then, I think na-mention yung biotechnology. So we have this stainless steel, no, used in propagating the cells na. So for large-scale production of vaccines, this is how they are being processed na. No? So it starts from small scale first and then shifting to large-scale production. So this is a very tight, no, closed system. It has to follow what you call GMP, good manufacturing practices. No? So aside from GLP, which we know na good laboratory practices here in a plant, they have to practice good manufacturing practices, because a single contaminant, no, one contaminant will destroy everything, no. So very controlled ito, the temperature, the pH, the humidity, and so on and so forth. It's very controlled, and they do a lot of screening, even filtration, checking for the presence or absence of a virus, no, etc. Yeah, and this is how they fill in the vaccines into individual syringes. No, so looking at this, it's fully automated. No, walang manual method dito, no, so that everything will be sterile. No, and then of course you have your packaging na as shown here. So they are available na in vials or in syringes, and yes, ito yung packaging system. Okay, so finally, this is my last slide. So for those who are actually allergic to eggs, no, then there's a hope, no. Like for example, the use naman of cell-based na flu vaccines. So this can be given to individuals like the NFL player, no, na allergic nga sa eggs, to have a cell-based, no. Yeah, and these cell-based vaccines are actually grown using animal cells naman, hoping na wala silang allergic naman sa animal cells. Yeah. Okay, sige. So that's my sharing, my additional inputs to everyone. All right. So yeah, we had a very fruitful evening tonight. All right. Sige, Peter and I'm sorry, Alex, paki-share yung slide na email mo sa akin so I can upload it in the TLC.
Yes, Doc.
All right. Okay, and then finally, I forgot to announce. So next week, one of your classmates won't be attending the class. So nag-ano na siya, according to Vincent. So she dropped already. So next week will be the report of one of your classmates in Sol. Tama? Yeah. So we will be listening to the report of Sol next week. Okay. So if there are no more questions, I think we're good. Gabriel, are you raising your hand? I can see.
Ayun, Doc. May follow-up question lang po sana.
Sure. Go ahead.
Sir, sorry. About the vaccine po. Kasi di ba po, lately we are mix-matching vaccines and boosters po.
Sa COVID, yeah, oo, totoo.
Is it advisable po ba na mag-mix and match? Pero recent studies naman po show na it is good naman po to extend.
Yes, oo.
Para ma-extend po yung immune response natin for the COVID infection.
Mm-hmm.
But there are vaccines po na, for example, Sinovac, you had your Sinovac vaccine. It is, parang meron po siyang certain na kino-consider na booster. Kunwari po, kasama po dun sa mga booster is Sinovac din po,
Mm.
and then Moderna, etc.
Mm-mm.
Gusto ko lang po sana tanungin, why po na other vaccines are not considered po dun? Kunwari po, sabihin natin, yung Sinovac nga po, hindi po siya, yung Pfizer na booster po, hindi po siya effective dun sa Sinovac po na main vaccine po.
Mm-hmm. Yeah. Thank you for that, Grace.
Thank you po.
Alex, would you like to comment first? Or can
You
it?
can help me. Regarding that, because the COVID vaccine right now is still under research. Remember, COVID was just in 2019, its outbreak, but the vaccine came out. So like fast track, where Sir Ken discussed that it takes actually years and years to develop a vaccine. Now, the World Health Organization, very controversial because I actually have the PDF of the Pfizer side effects, but that was not disclosed to some governments, and basta very controversial, Ma'am Gabi. And basta we listen to the World Health Organization and we listen to CDC. Yun lang po talaga yung masasabi ko.
So sorry, what is the question again of Ma'am Gabi?
Yeah, yung mixed vaccine, like let's say Sinovac, and then you have, let's say, mRNA like an Astra, or vice versa.
Well, I'll just listen to again what Sir Alex said, that we just need to listen to what the World Health Organization said, because apparently COVID vaccines are fast track since they were given emergency authorized use. Supposedly, vaccines take 10 to 15 years, but as we observe that vaccine development takes 6 to 8 months. Again, thanks to the prior MERS-CoV and SARS-CoV, which scientists have already studied their antigens and their possible use for vaccination, and then bang, pandemic happened. So mix and use, there's no possible side effects for mix and use vaccines.
Okay.
There's no possible adverse effects for mix and use of vaccines.
Thank you po.
Alright, sige. So if I may add, so my suggestion siguro, that would be a nice research topic. So in case if you have a subject like a research topic, you can use that problem, and you can address that problem by, let's say, doing a systematic review, and then later on you can proceed with a meta-analysis. So with that, because we know that systematic and meta-analysis are the strongest evidence, evidence-based. So we want to find out whether there's adverse effect or not, using this combination of vaccines. That will be very interesting. Yeah. So as sabi nga ni Alex, we need to gather pa more data because the COVID started in 2019, and as we all know, usually when we develop vaccine, it will take 10 years, 15 years to come up with a product that is safe and effective. So what we have now is, it's good, but we still do not know yet yung mga possible other side effects. Okay.
Thank you, Doc.
All right. Thank you, everyone. Do you have some other math questions or points you'd like to add? Josh, Jumela, Kirsten, Michaela, Joyce. Yeah, dalawa pa lang Joyce natin, one lang pala. Abdul Mahid, go ahead. You raise your hand.
Last, Doc. Why is that most vaccines, if not all vaccines, are developed and induced intramuscularly. Hindi ba
Ay,
pwede maging oral vaccine so that it would be easier
yeah,
for...
meron din mga oral and then may mga patch. Meron din subcutaneous. But yung conventional is IM, intramuscular.
Thank you, Doc.
All right. Okay. Anything else? I think I have that slide. No, if I can just share with you yung point ni Abdul Mahid. Last na lang, no? Kasi na-mention na naman, no? Since that was raised. So let me just pull that slide. I think I have a picture of that. Yeah, I saw it na. Wait lang ha. So let me just share this screen. Sorry, I wasn't able to change the background ha. But here, Abdul Mahid. So there are several ways of administrating vaccine. You can do ocular, like drops, nasal, oral immunization, vaginal, rectal immunization. Topical, like the adjuvants nga. Epidermal, ito yung patch. No? So these are some of the ways that we can administer the vaccine. I got this from this source. No? Yeah. Okay, sige. So I think it was a very fruitful... go ahead.
Regarding the question of Abdul Mahid, I guess because intramuscular site injection is that it optimizes immunogenicity as well as minimizing or reducing the adverse reactions at the injection site. That's why they usually inject it at intramuscularly.
Yes, correct. Tama. Alright. Mukhang ready na kayo mag-compre exam. Yeah, very good. Okay. Sige, so next week, we will hear another equally interesting and challenging topic. Yeah. Okay, sige, guys. See you next week. Thank you, everyone.
Thank you, Doc.
Thank you.
Thank you, Doc.
Enjoy your dinner.
Thank you, Doc.
Thank you, Doc.
Thank you too.